Eukaryotic RNA polymerase II transcribes all messenger RNA molecules in the cell. It is a huge complex of 10 different protein subunits. The active site of the enzyme lies at the interface between the two largest subunits. In this structure, a short stretch of a DNA-RNA heteroduplex was co-crystallized. New nucleotides would be continually added to the 3' hydroxyl group of the RNA shown in red. In the active site of RNA polymerase II, a single-stranded DNA template is transcribed into a complementary RNA transcript. The initial product is a DNA-RNA hybrid from which the newly synthesized RNA strand is stripped off. It leaves the polymerase via an exit groove on the protein surface. A pore on the backside of RNA polymerase II extends from the protein surface all the way to the active site. The nucleotide triphosphates used to build the growing RNA transcript enter the polymerase through this pore.